Hello everyone, myself Dr. Renu Solanki, Assistant Professor, Department of Geology, Deen Dayal Upadhyay College. In this video, I will explain about antigen-antibody interactions. Let me first define what are antigen and antibodies. Antigen is a foreign particulate matter or an allergen, we can say, which can bind to a specific antibody or T cell receptor. It is considered immunogen only when it is capable of eliciting immune response in the body. Antibodies are the proteins. They are also called as immunoglobulin, and they are produced by the plasma cells, which are differentiated B cells, and B cells are a type of WBCs. They are Y-shaped in structure as they have two heavy chain and the two light chain. So in this video, wherever you will see the Y-shaped structure, it means it is an antibody. Antigen and antibody interact with each other by means of non-covalent interactions, including hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals interactions, and you will see that these interactions are similar, which are used in the protein folding. There are some terms which are associated with the antigen-antibody interactions. First is the antibody affinity. It is defined as the strength of the total non-covalent interactions between a single antigen binding site on an antibody and a single epitope. Epitope is the part of the antigen where an antibody binds. Another term is antibody avidity. It is the strength of the multiple interactions between a multivalent antibody and antigen. If an antibody has low affinity and it has high avidity, so high avidity can compensate the low affinity. For example, in the case of the IgM, there are the 10 binding sites. So when an IgM binds to an antigen, it has the high avidity as compared to the IgG, where the binding sites are 2. The another term is the cross reactivity. Cross reactivity is when the antibodies are produced in response to an antigen, whereas they bind to another antigen having the similar properties, or we can say the similar epitopes. An example of the cross reactivity is ABO blood group antigen. We know there is the microflora in our gut, and in response to that, the antibodies are produced in response to the antigens which are present on the surface of these microorganisms in our gut microflora, and the antibodies are produced like anti-B, anti-A, or both the anti-A and B. So the cross reactivity occurs when a blood group A is and in a person having a blood group A, we transfused the blood blood group B. So in that it will have the antibodies B and the blood group A. They will cause the agglutination of the blood cells and it can be fatal. So the blood group typing is very important during the blood transfusions. Precipitation reactions occurs between an antibody and the antigen, but antibodies which result in the formation of the precipitates are referred to as the precipitations, and there are the two conditions in which the precipitation takes place. In antibody must be bivalent and antigen must be bivalent or polyvalent, then only they will form the lattice which will result in the formation of the precipitation. We can see over here, so these are the antigen and this is the epitope, and we see that in the polyclonal antisera there are the different antibodies which are specific to a particular epitope of the antigen. So the polyclonal antiserum result in the formation of a lattice, which is not possible in the case of the monoclonal antibodies, as the monoclonal antibody are specific to a particular epitope of the antigen. So for the lattice formation, or we can say for the precipitation, the antiserum should be polyclonal. Precipitation reaction can occur in the fluid as well as in the gel. On the basis of the precipitation reaction in the gels, there are some techniques. I will explain these techniques one by one. First is the radial immunodiffusion or Mancini method. It is a quantitative technique for determining the antigen concentrations. It is single immunodiffusion method because in this antigen or antibody diffuse, another remains static in the gel, that is the agarose. So let's see how the radial immunodiffusion occurs. So we will take the agarose. In that we will add the antibody when it is it is that much hot is able to touch, and in this what we do, we will make a well in the center and we add the antigen. In the well we will add the antigen, and antigen will diffuse, and in the zone of equivalence, zone of equivalence is the area where the amount of the antigen and antibody is such that excess of the antibody or antigen is not present, whereas all the binding sites of the antibody are blocked by the binding of the antigen. So this is the radial immunodiffusion. This slide is showing the radial immunodiffusion method. In this we made the gel. In the gel there are the antibodies. We bored the wells in the gel. After the solidification of the agarose, then we added the antigen, different concentration of the antigen, and depending on the amount we can see the precipitation line is formed. So by using the standard antigen we can form we can make a standard graph, and the amount of antigen can be found in the unknown sample by extrapolating from it. Another technique is the double immunodiffusion. As the name is indicating that it is double immunodiffusion, so in this method both the antibody and antigen will diffuse, whereas in the case of the radial immunodiffusion or the single immunodiffusion, only the antigen is diffusing. So there are the three different patterns that we observed in the case of the double immunodiffusion identity. Because in this what we used to do, in one sample, in one well we add the antibody, in the two wells, in these two wells we add the antigens. So we can compare the antigens whether they have the similar epitopes or not. If they have the similar epitopes, then we will get this type of the pattern, that is the identity and arc type. It means that the antigen have the similar epitopes. Second is the partial identity. In this there is an arc type. So in antigens were diffused in the two wells and antibody was there. The similar antigens, similar epitopes of the different antigen will give this arc, whereas this spur is because of the different epitopes on the antigen. And the third is the known identity, where the antigens having the different epitopes and the precipitation line cross each other. So this technique is used to find out or to compare the antigens. So we can see this in this video also. So in the double immunodiffusion, in the agarose in the agarose gel, two wells are poured, antigen and antibody are loaded, they diffuse, and in the zone of equivalence they form a precipitation line, and depending on the antigen we used, we will get the three different patterns. The third technique is the immunoelectrophoresis. It was developed by the Peter Nikolaevich Graeber, who was a French immunologist. It is a combination of the two techniques, that one is the electrophoresis and the second is the immunodiffusion. And the immunodiffusion over here is the double immunodiffusion. In the first part, what we used to do, that we make an agarose gel and we will pour it on the slide. After solidification, we will bore a well. In that we will load the antigen. Either we can make the well on the one side of the slide or in the center, because if we will load the antigen in the center, so we can separate the different components of the antigen, because the antigens can be the mixture of the different proteins. So depending on the charge of the components, the components of the antigen will get separated. Positively charged particle will move toward the negative electrode, and the negative charged particle will move toward the positive electrode. So after the separation of the components of the antigen, two troughs were cut in the gel, and in that we load the antibodies and we let the diffusion to occur. So it is the double immunodiffusion. So both the antigen and the antibody will diffuse toward each other and will form a precipitation line, where there is the matching of the antigen and the antibody. So let's see it in this technique, in this video. So the antigen is loaded over here. Components get separated according to the chart. And trough is made over here, and in that the antibody was diffused, and after the diffusion they will form the precipitation line in the zone of the equivalence. So this was about the immunoelectrophoresis. So in this video I explained about the antigen antibody interactions. There are certain terms related to the antigen antibody interaction, that is the avidity, affinity, cross reactivity. Then I explained about the what are the criteria that will be fulfilled for the precipitation to occur in the fluid as well as in the gel. Then I explained about the techniques, that is the radial immunodiffusion, double immunodiffusion, or Ottoloni method, or immunoelectrophoresis. A very thanks to all of you.